This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. We usually see viruses as external enemies, but they are actually the silent architects of the human story. In this narration, we'll explore how ancient infections shaped our genome, built our organs, and even directed human migration. We'll dive into the coevolutionary arms race between our cells and these microscopic invaders, and look toward a future where technology begins to outpace natural selection. Join us as we discover why we aren't just surviving viruses, we are made of them. Let's explore the viral evolution of humanity. Chapter 1: The Viral Architects of the Human Genome. This exploration into the relationship between viruses and human evolution begins with a fundamental shift in how we perceive ourselves. For a long time, the narrative of human history was written through the lens of bones, tools, and migrations. We viewed viruses solely as external predators, microscopic invaders that occasionally thinned our numbers but remained distinct from our biological identity. However, recent scientific research has pulled back the curtain on a much more intimate and ancient partnership. We are beginning to understand that we are not entirely human, at least not in the way we once thought. Our very genetic code is a patchwork quilt, and a significant portion of that fabric was woven by the very viruses we have spent millennia trying to outrun. Recent genomic analysis has revealed that viruses are responsible for a staggering 30% of all protein adaptations in the human lineage since we diverged from chimpanzees. This is not a marginal influence, it is a primary driver of our biological architecture. To understand how a virus can become an architect of the human body, we have to look at the mechanics of infection through a different lens. Most of us are familiar with how a common cold or a flu works. A virus enters a cell, hijacks its machinery to make copies of itself, and eventually bursts out to find new victims. For many viruses, the story ends there. But there is a specific category known as retroviruses that operates with a much more sophisticated and permanent strategy. These viruses, which include modern examples like the human immunodeficiency virus, or HIV, do not just use the cell, they integrate their own genetic material directly into the DNA of the host. Using an enzyme called reverse transcriptase, they convert their viral RNA into DNA and then stitch that sequence into the host's chromosomes. Once it is there, the virus becomes a permanent part of that cell's operating instructions. Under normal circumstances, this is a disaster for the individual cell, but it has no impact on the evolution of the species. If a retrovirus infects a lung cell or a skin cell, that genetic change dies when the person does. However, every once in a while, over millions of years, a retrovirus manages to find its way into a germline cell, the sperm or the egg. When this happens, and that cell goes on to create a child, the viral DNA is passed down to every single cell in that child's body. It is no longer an external infection, it is an inherited trait. These sequences are known as endogenous retroviruses, or ERVs. They are molecular fossils, the preserved remains of infections that our ancestors survived millions of years ago. The scale of this viral presence in our bodies is staggering. Current genomic mapping shows that roughly 8% of the human genome is made up of these endogenous retroviruses. To put that in perspective, the genes that actually code for the proteins that build our muscles, organs, and bones make up less than 2% of our DNA. We carry 4 times more viral code than we do traditional human genes. For decades, scientists looked at these massive stretches of repetitive virus-derived sequences and labeled them junk DNA. But as our ability to sequence and analyze the genome has advanced, we have discovered that this junk is actually a sophisticated control system.
The formal scientific term for this repurposing is exaptation, the process by which a trait evolved for one purpose is captured by evolution for an entirely different function. The human genome is not just a list of parts, it is a complex network of switches and regulators. Research into the deep history of our species has shown that viruses are the primary providers of these switches. A landmark study from the Pasteur Institute in 2025 and 2026 utilized CRISPR technology to silence specific endogenous retroviruses to see what would happen. The results were transformative. Researchers found that over 60 critical immune regulatory genes, including essential components of interferon signaling, are directly controlled by viral enhancers. These ancient invaders have become double agents, repurposed by evolution to serve as our primary sentries. When our body detects a danger signal, these viral switches flip on, coordinating a massive cellular defense that would be impossible without them.
This viral architecture extends into the very structure of our brains. Research comparing the human genome to that of our closest living relatives, the chimpanzees, has revealed a striking difference in a gene called PRODH. This gene is involved in the wiring of the brain, specifically in the hippocampus. While both humans and chimps possess this gene, it is significantly more active in the human brain. The reason for this difference is a viral switch. Millions of years ago, a virus hopped into a specific location next to the PRODH gene in one of our ancestors. This did not happen in the lineage that led to modern chimpanzees. This single viral insertion changed the way that gene is expressed, potentially altering the cognitive wiring and synaptic density that separates us from other primates. The process of harnessing these viruses is not a passive one. It is the result of an intense, millions of years long arms race. Our cells have developed specialized proteins called KRAB zinc finger proteins, which act as virus tamers. There are over 300 different types of these proteins in the human genome, and their job is to seek out viral sequences and pin them in place. Originally, these proteins evolved to silence the viruses and prevent them from causing harm. But over time, the relationship shifted from suppression to domestication. Instead of just keeping the viruses quiet, these proteins began to recruit them, using the viral DNA to help regulate other human genes. This is a highly refined way of managing cellular activity, and it explains why different species have such unique patterns of these silencing proteins. As we look deeper into the history of life, the boundary between what is a virus and what is a host begins to blur. We traditionally think of life as a tree with distinct branches, but the reality is more like a web. Horizontal gene transfer, where genetic material moves between unrelated species via viral vectors, has been a constant force in our development. This challenges our very definition of what it means to be alive. Even at the molecular level, the similarities are striking. This suggests that the first viruses may have been gene thieves, capturing proteins from their hosts only for the hosts to eventually steal them back. The discovery of giant viruses, like the Mimivirus and the Pandora virus, has further complicated this picture. These viruses are so large and possess such complex genomes that they actually rival some bacteria. They carry genes for metabolism and protein synthesis, functions that were previously thought to be the exclusive domain of cellular life. They even encode their own complexes for translating genetic information, giving them an unprecedented level of autonomy from the host cell. Some scientists hypothesize that these giant viruses are the descendants of more complex, free-living ancestors that chose a parasitic path and shed their unnecessary genes over time. This suggests that viruses are part of an evolutionary continuum that predates the last universal common ancestor of all modern life. If we accept that viruses are the primary architects of our genome, we have to reconsider the story of human survival. We are not just the survivors of ancient plagues, we are the products of them. Viral selection may even be responsible for the loss of the N-glycolineuramic acid, or N5GC, cell surface receptor, a major evolutionary divergence that separates humans from other primates and changed our susceptibility to various pathogens. Every time a new virus swept through a population of our ancestors, it acted as a selective sieve. Those who survived often did so because their genomes were flexible enough to co-opt the invader's tools. While the acquisition and expansion of endogenous retroviruses have slowed down significantly, diminishing fourfold in hominids compared to other primates over the last 30 million years, the process remained active until very recently. The virus known as HERVK, HML2, remained active and capable of infecting the germline until approximately 200,000 years ago, leaving lineage-specific copies in humans, Neanderthals, and Denisovans. We can see this process happening in real time today in other species. In Australia, the koala population is currently undergoing a genome invasion by a retrovirus. We are watching as a virus moves from an external infection to a permanent inherited part of a species' DNA. By studying these events, we are gaining a clearer understanding of how our own ancestors were transformed.
This genomic architecture provides us with a new way to look at modern disease. When we see faults in genes like PRODH, we see their links to brain disorders. When we see dysregulation in viral switches, we see the roots of autoimmune diseases and cancer. The same ancient elements that give us our strength and our unique human traits also represent our greatest vulnerabilities. Understanding the logic of virus evolution is no longer just an academic pursuit. It is a requirement for the future of human health. We are moving toward a world where we can manipulate these cellular functions instead of just responding to symptoms. As we move forward, we must ask ourselves how much of our behavior and our physical form is truly our own. If a viral switch in our brain influences how we think, and a viral gene in our immune system determines how we survive, where does the virus end and the human begin? We have spent millions of years domesticating these microscopic invaders, turning their weapons into our tools. But the process of architectural change is never truly finished. This leads us to consider not just the switches that regulate our genes, but the very organs that allow us to reproduce and flourish. Structures that were not merely influenced by viruses, but were physically constructed from their stolen parts.
Chapter 2. Borrowed Architecture, the Viral Scaffolding of the Human Body. If the human genome is a blueprint, as we explored previously, then the organs of our bodies are the finished rooms of the house. But as we look closer at the construction of these rooms, we find that some of the most critical structural components were not designed by our own ancestors. Instead, they were borrowed from the very viruses that once threatened to wipe us out. As we saw in the previous chapter, the process of capturing viral tools and repurposing them for life-sustaining functions is the reason we exist in our current form. Without a series of chance infections that occurred tens of millions of years ago, the way we are born and the way we look would be fundamentally unrecognizable. The most dramatic example of this viral borrowing is the placenta. This organ is the essential bridge between mother and child, but its existence requires solving two seemingly impossible biological problems. First, the mother's immune system must be prevented from attacking the fetus, which it should technically recognize as a foreign invader. Second, the fetus needs to fuse itself to the mother's blood supply to draw nutrients without the two systems fully mixing. The solution was provided by an ancient retrovirus. Researchers have identified a specific human gene called syncytin, which is active only in the placenta. Its job is to create a molecule that allows cells to fuse together into a single, seamless layer of tissue known as a syncytium. This layer acts as a sophisticated barrier, a selective filter that manages the exchange of gases while acting as an immunological cloak, hiding the fetus from the mother's killer T cells. This is not a human gene. It is a viral one. In its original life as part of an infectious retrovirus, this gene coded for an envelope protein designed to allow the virus to fuse its membrane with a host cell. Millions of years ago, our ancestors captured this fusion tool and used it to build the placental barrier. We can even date the specific fossil of this transition. On human chromosome 17, there is a specific endogenous virus that is at least 104 million years old. This marker is found only in placental mammals, providing a clear genetic timestamp for when our ancestors began using viral machinery to move away from laying eggs. What makes this story even more incredible is that it didn't just happen once. Evolution has a habit of returning to the viral toolbox whenever it needs to build or refine a placenta. While the original capture likely happened 150 million years ago, the record suggests that repeated subsequent infections overwrote the original genes. This explains why different mammals have different syncytin genes today. Humans and primates share two specific versions, while mice, cats, and dogs have entirely different viral genes that perform the exact same function. They each caught different viruses at different points in their evolutionary history to solve the same structural problem. For a long time, this was a compelling theory, but scientists lacked a smoking gun, an example of a species caught in the middle of this transition. That changed with the study of the Mabuya lizard. Most reptiles lay eggs with hard shells, but certain species of these lizards in the Andes Mountains have made a radical shift. They give birth to live young using an advanced placenta remarkably similar to our own. When geneticists analyzed their DNA, they found a gene essential for this placenta that came from a virus. The acquisition of this viral DNA coincided exactly with the lizard's shift from being an egg-laying species to a placental one, confirming that viruses are the primary catalysts for the creation of new organs. This logic of borrowing extends to the very hallmark proteins that define what a virus is. We now know there is a massive, ancient exchange of parts between viruses and cellular life. For instance, the jelly roll fold, a specific structural shape found in the protein shells or capsids of a vast array of human-infecting viruses, has a close relative in the human tumor necrosis factor TNF family. These are proteins our bodies use for cell signaling and immune responses. This structural similarity suggests a deep history of theft and counter-theft, where the proteins used to build a virus's protective shell were repurposed to build the signaling systems of our own cells. Beyond these large-scale structures, viruses act as the architects of our physical appearance. Recent research into the viral switches in our genome has revealed that these elements are responsible for the differences in how human faces are built in the womb compared to those of chimpanzees. While we share the vast majority of our protein-coding genes with chimps, the way those genes are turned on and off during development is governed by viral enhancers. These switches dictate the shape of the jaw, the prominence of the brow, and the alignment of the nose. We do not look like our primate cousins because ancient viruses essentially rewrote the regulatory manual for the human face. This regulatory influence extends into the very cells that allow us to grow. Stem cells, the blank slate cells found in developing embryos, have the unique ability to become any type of tissue in the body, a state known as pluripotency. Recent research has shown that a family of retroviruses known as HERV-H is essential for this process. These viral sequences do not code for proteins. Instead, they are copied into molecules of RNA that act as managers. If these viral sequences are suppressed, stem cells lose their flexibility and begin to differentiate prematurely. In a very real sense, viral elements ensure we have a large enough pool of undecided cells to build complex structures like the human brain. But our viral history shapes more than just the physical architecture of our organs. It also defines the brain of our immune system. To understand why two people can be exposed to the same virus and have completely different outcomes, we must look at the molecular diary etched into our immune cells through epigenetic modifications, small chemical tags that tell the body which genes to turn on or off. These life experiences actually rewrite the way our viral genes behave. While some of our immune factory settings are inherited, others are shaped by experience. When you survive an infection, your body recalibrates the viral switches in your genome, fine-tuning how your immune cells will react to future threats. This epigenetic fingerprint means that your history of viral infections is essentially a training manual for your survival, stored within the very viral elements that once sought to exploit you. We see the power of this internal viral machinery in other species as well. Domestic sheep are often plagued by a retrovirus that causes a form of lung cancer. However, some sheep have integrated a copy of this virus into their own DNA. Curiously, these sheep are now immune to the circulating version of the virus. The internal domesticated virus blocks the receptors that the external virus tries to use to enter the cell. It is a form of genetic vaccination that the species performed on itself over thousands of years, turning a predator into a permanent internal guard. This brings us to a fundamental shift in how we understand the self. We are not just a collection of human genes that occasionally get sick. We are a sophisticated hybrid. Our ability to carry a child to term is a viral trait. The shape of our faces and the flexibility of our stem cells are governed by viral switches. Even the way our immune system remembers a past infection is managed by the remnants of ancient invaders. We have taken the machinery of death and used it to power the machinery of life. But if our biology is so dependent on these ancient borrowed genes, what happens when the environment changes? If our survival has always been dictated by our ability to absorb new pathogens and repurpose their tools, how did these microscopic directors shape where our ancestors could live and how they moved across the planet? This leads us to look beyond the individual body and toward the history of the entire human species. How did the viruses we carried and the ones we encountered act as the silent directors of human migration? Chapter 3. The Invisible Walls. How Pathogens Mapped Human History. When we look at a map of the ancient world, we see the rise and fall of empires, the crossing of land bridges, and the slow spread of our ancestors across every corner of the globe. We often attribute these movements to the search for food, the changing climate, or the pressure of rival tribes. But recent scientific research has begun to reveal another, far more subtle force that has dictated where humans could live, where they had to flee, and who survived the journey. Pathogens have acted as the silent directors of human history, serving as invisible walls that cordoned off entire regions and as selective sieves that reshaped the genetic identity of those who dared to cross them. To understand the story of human migration, we must look not just at the tracks left by ancient feet, but at the microscopic trail of the diseases that followed them. For tens of thousands of years, one of the most powerful map makers on the African continent was not a human leader, but the malaria parasite. New biodiversity modeling and climatic reconstructions from the Max Planck Institute for Geoanthropology and the University of Cambridge have provided a startlingly clear picture of how this disease influenced the distribution of early humans. By mapping the historic habitats and climatic envelopes of the three primary mosquito species within the Anopheles genus responsible for transmitting malaria in Africa over the last 70,000 years, scientists have discovered a striking pattern of avoidance. For the vast majority of our early history, the places where humans chose to live almost never overlapped with the regions where malaria was most prevalent. This wasn't a coincidence. It was a survival strategy. These ancient ancestors likely viewed certain lush, water-rich environments as death traps, even if they didn't understand the microscopic reason why. This avoidance had profound implications for how human populations interacted.
Because they were effectively forced into specific ecological niches to avoid the parasite, different groups of humans were often isolated from one another by malaria barriers. This prevented the exchange of ideas, technology, and genes, contributing to the complex, mosaic-like evolution of our species across the continent. The parasite essentially dictated the borders of human territory, creating a fragmented landscape where survival depended on staying within the safe zones defined by mosquito ecology. However, this pattern of avoidance shifted at two critical junctures in our history. The first occurred roughly 60,000 years ago, coinciding with the successful migration of modern humans out of Africa. As populations moved and environmental conditions changed, the overlap between human settlements and malaria-prone regions increased. This suggests that the very act of leaving the continent required our ancestors to run a gauntlet of disease, potentially selecting for individuals with more robust innate immune responses. The second major shift happened about 12,000 years ago, as humans began to abandon their nomadic lifestyles for permanent settlements. The transition to agriculture and sedentary living forced humans to stay in one place, regardless of the seasonal rise of mosquito populations. This was the moment when we stopped running from the parasite and began the long, deadly process of living alongside it.
This shift to permanent settlement didn't just change where we lived, it fundamentally altered our biological trajectory. When a population is penned down in a high-disease environment, the pressure of natural selection becomes intense and rapid. As we will explore later in the context of the modern coevolutionary arms race, viruses like HIV continue to act as genetic sieves in places like South Africa, rapidly favoring protective immune variants in those who survive. This is a vivid reminder that the invisible walls of the past were often reinforced by the deaths of those who lacked the right genetic tools to survive. The relationship between migration and disease is also written in the genomes of the pathogens themselves. The field of paleovirology has made incredible strides in recovering ancient viral nucleic acids from human remains, allowing us to reconstruct the history of epidemics that were previously only known through vague historical accounts. We have now sequenced the genomes of roughly 200 ancient human viruses, including hepatitis B, smallpox, and measles. These molecular fossils are changing our understanding of the timeline of human survival. For example, it was long believed that measles was a relatively modern disease, appearing only after the rise of large, dense cities. However, new models that account for long-term purifying selection suggest that measles actually diverged from its animal relative, rinderpest, as early as the 6th century BCE. This earlier date maps onto a period of increasing human-animal proximity and the expansion of trade networks. It tells us that as our ancestors moved and brought their livestock with them, they were inadvertently creating the conditions for new, devastating pathogens to jump the species barrier. These viruses didn't just follow human migration. They often arrived first, clearing the way or wiping out entire populations before they could establish a foothold in a new territory. This microbial traffic is particularly evident in regions that have historically served as crossroads for trade and nomadic movement. In northwest China and adjacent countries like Kazakhstan and Pakistan, the diversity of tick-borne viruses is staggering. Recent meta-transcriptomic sequencing of nearly 10,000 ticks in this vast geographic area identified 92 different RNA viruses, including 10 that are known to infect humans and other mammals. This region, a vital artery of the ancient Silk Road, is a hotspot for pathogens like the Crimean Congo hemorrhagic fever virus and the newly discovered Tacheng tick viruses. The high prevalence of these diseases is directly linked to the large populations of livestock, sheep, cattle, and camels that have been moved across these lands for millennia. These animals act as mobile reservoirs, carrying the ticks and their viral cargo along ancient trade routes. For the people living in these regions, survival has always been a matter of balancing the economic necessity of livestock with the constant risk of viral spillover.
This has created a unique disease ecology where the risk of infection is not just a seasonal event, but a permanent feature of the landscape, dictated by the movement of trade and the migration of nomadic tribes. Whenever humans migrated into a new territory, cleared a forest for agriculture, or traded in a new type of animal, they were essentially rolling the biological dice. Migration into new territories increased exposure to pre-armed viruses, pathogens that already possessed the capacity to infect human cells while circulating in their natural animal reservoirs. Most of the time, the encounter resulted in a dead-end infection. But occasionally, a virus found itself in a human host with the right genetic vulnerabilities. If that individual was part of a migratory group or a growing settlement, the virus had the perfect conduit to spread.
The impact of these pathogens extends even to our most basic biological functions, such as how we digest the food we find in new environments. While we previously touched on how diet can drive genetic change, it is important to recognize that these adaptations often occur in the context of disease. The Andean populations that adapted to a potato-rich diet did so in an environment where they were also facing unique pathogens like the Andes virus, a type of hantavirus carried by long-tailed pygmy rice rats. In this region, the Quechua people possess an average of 10 copies of the AM1 gene, the highest number of any population globally. This specific detail makes the evolutionary sieve argument concrete. The ability to efficiently process energy from starch wasn't just about general nutrition. It was about fueling the immune system to survive a landscape of persistent viral threats. In the Andes, potato power was the metabolic fuel required to survive a hantavirus infection. This perspective forces us to reconsider the very nature of human survival. We often think of our ancestors as the protagonists of their own story, making conscious choices about where to go and how to live. But in many ways, they were like particles in a fluid, being pushed and pulled by the invisible currents of the virusphere. A tribe might have moved north not because the hunting was better, but because the malaria wall to the south had become impassable. A civilization might have collapsed, not because of a failure of leadership, but because a new trade route brought a virus that their immune systems weren't prepared to handle. Even the resources available to ancient coastal populations were dictated by viral activity. In the oceans, a phenomenon known as the viral shunt plays a critical role in the ecosystem. Marine viruses are responsible for the lysis, the bursting, of a significant portion of microbial cells every day. This process recycles organic matter back into the water, supporting the very base of the food chain. Without this viral activity, the nutrient cycles that support fish and shellfish populations would collapse, fundamentally altering the migration patterns of coastal human groups who relied on the sea for survival. We are now entering an era where we can see the full extent of this influence. We can map the regional structuring of viruses across continents, from the high viral diversity in Africa to the specialized clades that dominate in Europe and Asia. We are beginning to understand that pathogens were not just obstacles to human progress, they were the architects of our distribution. The map of human genetic diversity is, in many ways, a map of historical disease outbreaks. The reason a person in one part of the world has a different immune profile than someone in another is often because their ancestors survived different viral sieves. These microscopic directors have dictated the terms of our survival for millions of years, deciding which lineages would flourish and which would vanish into the archaeological record. But as we look at the way these viruses have shaped our past, we must also look at how they are continuing to change in the present. We have seen that viruses don't need to adapt to us to make the jump. But once they arrive, the clock starts ticking. They begin a rapid process of fine-tuning, searching for the mutations that will allow them to spread faster and evade our defenses. This raises a critical question about the nature of our relationship with the microscopic world.
If the jump from animal to human is often just an unfortunate coincidence of exposure, what are the specific factors that allow a virus to suddenly turn from a rare, sporadic infection into a global pandemic?
Chapter 4. The Unfortunate Coincidence. We often imagine the birth of a pandemic as a calculated move by nature, a slow and deliberate process where a virus carefully sharpens its tools to pick the lock of the human immune system. In this traditional view, a pathogen circulating in a bat, a bird, or a primate must undergo a long series of specific mutations, perhaps through a period of stuttering transmission, where it fails to spread effectively but gradually learns the human language, before it can finally spark a global crisis. We have long assumed that these viruses are evolutionarily special, that they have been pre-adapted through a grueling gauntlet of natural selection to become human killers. However, recent large-scale genomic research has begun to dismantle this narrative. What we are finding is something much more unsettling. Most of the viruses that have caused our most devastating modern outbreaks did not need to adapt to us at all. They made the jump not because they changed, but because of an unfortunate coincidence.
To understand how this works, we have to look at the evolutionary record left behind in the genes of viruses like Ebola, Marburg, and the various coronaviruses that have emerged in the 21st century. By using sophisticated phylogenetic frameworks, scientists can now peer into the stem branch of a virus's family tree. The period of time between when a virus existed solely in its animal reservoir and when it first established itself in the human population. By using sophisticated phylogenetic frameworks, scientists can now peer into the stem branch of a virus's family tree. The period of time between when a virus existed solely in its animal reservoir and when it first established itself in the human population. If the traditional model of pre-adaptation were true, we would expect to see a massive shift in the intensity of natural selection during this period. We would see positive selection, where advantageous mutations are being rapidly fixed in the viral genome as it prepares to enter a new host. But when researchers analyzed the genomes of Ebola, Marburg, Mpox, and SARS-CoV-2, they found that the selection pressures on these viruses remained completely unchanged until the very moment they began spreading between humans. This discovery suggests that these viruses already possess the basic biological keys required to infect human cells and sustain human-to-human transmission while they were still circulating in their natural animal hosts. They were, in effect, pre-armed for us by accident. This implies that the biological world is teeming with viruses that are already capable of causing human epidemics. They simply haven't had the opportunity to do so yet. The barrier between us and a new pandemic is not a lack of viral adaptation, but a lack of exposure. This shifts the focus of global health from watching how viruses change to watching how we interact with the natural world.
However, the genomic record also shows that this unfortunate coincidence is not the only way a virus can enter our species. Research by Havens and colleagues in 2026 has highlighted a crucial exception that proves the rule: the original SARS-CoV virus of 2003. Unlike its successor, the 2003 virus actually did show a significant change in selection intensity before it became a major human threat. It underwent a period of rapid adaptation in an intermediate host, specifically the palm civet, where it refined its ability to bind to the receptors it would eventually use to infect humans. This contrast is vital. It shows that while the traditional model of adaptation in an intermediate host still exists, it is no longer the primary way we see modern pandemics emerging. The viruses that catch us most off guard are often the ones that require no such apprenticeship. The case of SARS-CoV-2 provides perhaps the most striking evidence for this new understanding. Despite the intense controversy surrounding its origins, the genomic record shows that the selection signatures on the virus were essentially identical to those found in its natural animal reservoirs right up until its emergence in the human population. There was no detectable period of prolonged evolution in an intermediate host that shaped its ability to infect us.
It arrived in the human population ready to go. The virus's spike protein was already capable of binding to the human ACE2 receptor with high affinity, a trait that likely evolved for a completely different purpose in its original host but worked perfectly on us. It was only after the virus was firmly established in humans that we began to see a period of rapid adaptation begin, with the emergence of variants that fine-tuned the virus's ability to spread and evade our immune systems. This pattern is consistent across multiple viral families. In the Filoviridae family, which includes Ebola and Marburg, the story is the same. During the 2013 to 2016 West African Ebola epidemic, we did see the virus eventually mutate to increase its tropism for human cells, making it better at infecting us than it was at infecting its original animal host. But those mutations happened during the outbreak, not before it. The initial jump was made by a virus that was evolutionarily indistinguishable from the versions circulating in bats. These pathogens are not waiting to become dangerous to us. They are dangerous to us by default.
This research has also provided us with a powerful new tool for forensic virology, the ability to distinguish between a natural spillover and a laboratory-linked event. While natural zoonotic viruses show no change in selection intensity before they hit humans, viruses that have been passaged in a laboratory or through artificial cell cultures leave a distinct genetic signature. In a laboratory environment, a virus is often released from the intense purifying selection it faces in the wild. It doesn't have to worry about a host's complex immune system or the need to transmit between individuals. This results in relaxed selection, where mutations that would normally be weeded out are allowed to persist. This is the inverse of the natural coincidence. Instead of a virus being pre-armed by the rigors of the wild, it is softened by the comforts of the lab. We can see this clearly in the history of the H1N1 influenza virus. While most pandemics show a steady, natural accumulation of mutations, the reemergence of H1N1 in 1977 was a bizarre outlier. The virus that appeared in 1977 was nearly identical to a strain that had circulated in the 1950s, as if it had been frozen in time for 2 decades. When scientists analyzed its selection regime, they found a pattern consistent with laboratory passage, supporting the long-held theory that this particular pandemic was sparked by an accidental release during a vaccine trial. By establishing what a normal natural spillover looks like at the genomic level, we now have a benchmark to identify when something has gone wrong in a controlled environment.
The unfortunate coincidence isn't just about the virus being able to enter our cells. It's also about the specific vulnerabilities of the human population at the moment of contact. While genomic research hasn't found evidence of prolonged stuttering adaptation prior to known outbreaks, the mechanism of stuttering transmission remains a valid way for a virus to find its footing once it arrives. We are beginning to understand that certain individuals may act as mutation incubators or superspreaders because of their own genetic makeup. For instance, people with inborn errors of immunity, particularly those involving interferon signaling, may be unable to clear an initial infection quickly. This gives a virus that has just made the jump more time to replicate and explore the human sequence space, potentially finding those first few critical adaptations that turn a dead-end infection into a transmissible one. In this scenario, the virus doesn't need to be pre-adapted to the whole species. It just needs to find one person whose lock is already broken.
This dynamic is further complicated by the way viruses interact with one another during these initial jumps. We often study a single pathogen in isolation, but in the real world, co-infection is the norm. In mixing vessels like pigs that carry influenza, multiple strains of a virus often infect the same cell at the same time. This allows for reassortment, where viruses swap entire segments of their genomes. This is a form of modular evolution that can create a hybrid virus with unique properties overnight. The 2009 H1N1 swine flu pandemic was the result of a series of such swaps between human, bird, and pig viruses that had been occurring for years.
The final virus didn't need a long period of fine-tuning. It was a Frankenstein pathogen, assembled from parts that were already functional in their respective hosts. The sheer diversity of the virus sphere means that the number of these unfortunate coincidences is likely much higher than we realize. The complexity of viral life suggests that the rules of what can and cannot infect a human are constantly being rewritten. They aren't just passive packets of RNA, they are active participants in an evolutionary continuum. This perspective challenges the very core of our pandemic preparedness strategies. If we continue to wait for viruses to show their hand by adapting before an outbreak, we will always be one step behind. We have been looking for signs of a virus learning to infect us when we should have been looking at the fact that many of them have already graduated.
We must also consider how our own evolutionary history has prepared the ground for these coincidences. As we saw in earlier chapters, our shared molecular language with viruses, the result of millions of years of integrations that built our very biology, creates a home field advantage for many zoonotic pathogens. Because our genomes are so heavily built from viral parts, animal viruses often find they are not entering a foreign environment, but a system that was, in many ways, designed by their ancestors. As we move forward, we have to ask, if the jump to humans is often an accident of exposure rather than a feat of adaptation, what happens once the virus is inside? How does the unfortunate coincidence transform into a permanent co-evolutionary arms race? We have seen that the virus doesn't need to change to get in, but once the first human-to-human transmission occurs, a clock is triggered. The virus begins to feel the pressure of the human immune system, and the human immune system begins to feel the pressure of the virus. This leads us to a much more complex and interactive phase of our history. We are no longer just looking at a virus that happened to fit a lock. We are looking at a system where the lock and the key are both constantly changing shape in a desperate attempt to outmaneuver one another. If a virus can jump to us without adaptation, can it also influence our behavior to ensure its own survival once it arrives? Chapter 5: The Red Queen's Race and the Behavioral Battlefield. The relationship between humans and viruses is often described as a co-evolutionary arms race, a term that captures the relentless back-and-forth struggle for dominance. In biological terms, this is frequently referred to as the Red Queen hypothesis, named after the character in Lewis Carroll's Through the Looking Glass, who remarked that it takes all the running you can do just to stay in the same place. For every defensive innovation our immune system develops, viruses eventually find a countermove. And for every viral strategy to infiltrate our cells, our bodies eventually build a new wall. This chapter explores the front lines of this ancient conflict, moving from the staggering genetic diversity of our immune systems to the unsettling possibility that viruses are manipulating our very behavior to ensure their survival and spread. The most visible battlefield of this arms race is located within a specific region of the human genome, known as the human leukocyte antigen, or HLA, complex. Also referred to as the major histocompatibility complex, or MHC, this is the most variable part of our entire genetic code. The HLA complex acts as the training ground for our immune system, producing proteins that grab fragments of invading viruses and present them to our T cells. Because viruses mutate so rapidly, our species has had to maintain an incredible level of diversity in these HLA genes. If we all have the same immune training manual, a single viral mutation could render our entire species defenseless. Instead, we possess thousands of different HLA variants, ensuring that even in the face of a devastating pandemic, there will always be individuals whose immune systems happen to have the right tools to recognize the threat. Viruses have developed a mirror image strategy to stay ahead of this adaptation, known as antigenic evolution. Some viruses, like measles, are relatively stable, possessing a single molecular lock that rarely changes. But others, like the seasonal influenza A strain H3N2, possess what is essentially a combination lock that rotates constantly.
These viruses move through a coordinate system of shapes, an antigenic space, to stay ahead of the immune system's memory. By analyzing an atlas of viral adaptation, researchers have found that this continuous movement through shape space is far more common than we once realized. Norovirus, respiratory syncytial virus, and certain seasonal coronaviruses all show high rates of adaptation in their receptor binding proteins, ensuring that the antibodies we built during a previous infection are no longer effective. In the case of SARS-CoV-2, we can see the forensic specifics of this race. Mutations like D614G emerged early to enhance transmissibility, while others like N501Y tightened the virus's ability to bind to our receptors. This optimization is often inadvertently driven by our own defensive mechanisms. One of our primary lines of innate defense involves a family of proteins called ApoBe. These proteins are genome editors that physically attack viral RNA or DNA, inducing massive numbers of mutations to break the virus's genetic code. While this editing is intended to destroy the virus, it also creates a massive pool of genetic variation. In a population of billions of viral particles, some of these host-induced changes might actually be beneficial, providing the sparks for the virus's next evolutionary leap.
This continuous adaptive evolution is also evident in the human metapneumovirus, or HMPV. A specific lineage known as the A2B2 clade has become the dominant version of the virus globally by acquiring specific genetic duplications, specifically a 180 nucleotide duplication in its G gene. This structural change gave the virus a selective advantage in the modern human environment, a strategy remarkably similar to duplications seen in respiratory syncytial virus. These are not random shifts, they are calculated maneuvers in a high-stakes game of biological chess. This brings us to the most complex and perhaps most unsettling front of the arms race, the behavioral battlefield. For a virus, infecting a host is only half the battle. It must also ensure it can find a new host. While symptoms like coughing and sneezing are viral strategies for aerosol transmission, recent research suggests that viruses may be pulling much deeper strings. All infectious agents are under evolutionary pressure to manipulate host behavior to enhance their transmission. One way to do this is by suppressing sickness behavior. Normally, when we are ill, we feel lethargic and social withdrawal sets in, a host strategy to conserve energy and protect the community. However, many viruses reach their peak transmissibility a day or two before symptoms appear. By keeping the host feeling healthy and active during this window, the virus ensures that the individual continues their normal social routines. This is a form of silent exploitation. Our behavioral immune system naturally uses the emotion of disgust to keep us away from the sick. But viruses may evolve to reduce the outward signs of disease that trigger this disgust in others, allowing for stealthy spread through our social networks. There is even evidence that some viruses might increase host sociability during the peak transmission window. One study used the influenza vaccine as a proxy for infection and found that in the 48 hours following the injection, individuals actually interacted with significantly more people and in larger social groups than they did before. The mechanism for this behavioral manipulation often involves the disruption of interferon signaling. As we have seen, our genome contains dozens of viral switches that control these signals. While these switches are part of our architectural defense, viruses have evolved sophisticated ways to block or interfere with the resulting communication. By cutting the lines between the immune system and the brain, the virus prevents the host from realizing they should stay home. This viral influence on the mind may even be linked to more permanent shifts in human psychology, creating a tug-of-war over host activity levels that manifests as mood disorders. This co-evolutionary dance even influences how we interact with our environment. Consider the norovirus, whose ability to sweep across the globe is tied to our own genetics.
Susceptibility to the most common strains is determined by the FUT2 gene, which controls certain antigens on our intestinal cells. About 20% of people of European descent carry a mutation that makes them non-secretors, granting them natural resistance to common norovirus strains. This genetic polymorphism is likely the result of thousands of years of outbreaks and is closely linked to the historical migrations of coastal populations who relied on high-density social structures and maritime trade, environments where norovirus thrives. As we look at the totality of this arms race, we see that viruses have shaped not just our physical form, our social structures, and our mental health. We are a species defined by our high density and our intense social connectivity, traits that have allowed us to dominate the planet, but have also made us the ultimate prize for viral evolution. We have built an immune system of unparalleled complexity to defend this social nature, and in response, viruses have learned to navigate the very wiring of our brains to ensure that our sociality remains their primary engine of growth.
This realization forces us to ask a difficult question about the nature of our progress. For millions of years, this arms race was the primary driver of human biological change. Our genes were constantly being reshuffled, and our behaviors were being tuned in response to a never-ending barrage of viral threats. But in the last century, a new force has entered the arena. With the advent of vaccines, antivirals, and global surveillance, we have begun to interfere with the fundamental mechanics of this conflict. If we are no longer being shaped by the viral sieve, if we are using technology to freeze the arms race in its tracks, what does that mean for the future of human evolution? Chapter 6: The Technological Skin. For billions of years, the story of life was written in the language of survival and death. As we have explored, viruses were the primary editors of this text, using the viral sieve to decide which genetic traits would flourish and which would vanish. If a population lacked the right immune receptors to survive a plague, that lineage ended. If an individual possessed a rare mutation that blocked a viral entry point, they lived to pass that advantage to their children. This was the relentless, grinding machinery of natural selection. But in the last century, and specifically in the last few decades, something unprecedented has happened. For the first time in the history of our species, we have developed the tools to step out of this evolutionary line. By using vaccines, antiretroviral therapies, and advanced genomic surveillance, modern medicine is effectively halting the clock on human viral evolution. We are no longer waiting for our genomes to adapt to pathogens. We are forcing our technology to adapt instead. This shift is most visible in the transition from daily pills to what can be described as a technological skin, medical interventions that move beyond temporary treatments toward permanent biological shields. The emergence of long-acting injectables like Cabenuva, the first complete long-acting regimen for HIV-1, and Apretude, a long-acting injectable for PrEP, represents a radical departure from traditional medicine. These are not merely drugs, they are semi-permanent technological layers that decouple our survival from our innate genetic susceptibility. By providing months of protection in a single dose, we are effectively installing a synthetic immune system that operates independently of our inherited HLA variants. The consequences of this technological shield are already measurable in the global genome. In regions where infectious diseases once exerted massive selective pressure, that process has been effectively paused by the widespread use of modern medical interventions. We have traded a biological adaptation for a technological one, freezing the genetic diversity of the population in its current state. While this is a triumph for human rights and individual health, it creates a new kind of precariousness. Our biological memory is being replaced by a technological memory that is entirely dependent on infrastructure. We saw the fragility of this shield in 2025 and 2026, as shifts in global health funding led to clinic closures and supply chain disruptions. When the technological shield falters, the biological clock starts ticking again, but it does so in a population that has become entirely dependent on that shield for its survival. This dependency extends to how we manage the entire biosphere. We are moving toward a One Health early warning system, an integrated infrastructure that monitors human, animal, and environmental health as a single interconnected web. This is no longer a theoretical goal. It is a necessity driven by the way we have industrialized our environment. For example, the detection of H5N1 avian influenza in cattle milk and milking equipment has revealed that our evolution is now tied to the genomic surveillance of our food chain. We are not just monitoring human outbreaks. We are attempting to manage the viral load of the entire planet. Our survival now depends on our ability to detect a spillover in a dairy farm or a poultry market before it ever reaches a human host. This is the new frontier of natural selection. It is no longer about who has the best immune system, but who has the best sensors. The Red Queen race has moved from the theater of the human body to the theater of the research lab. We are running as fast as we can, but the running is now being done by bioinformaticians and molecular biologists, rather than by our own white blood cells. This race is being accelerated by the integration of artificial intelligence and machine learning in drug discovery. AI is now being used to predict how a virus will mutate before it even happens, allowing scientists to design augmented defenses that are pre-adapted to future viral variants. We are using silicon to outpace carbon, attempting to solve the problem of viral evolution through sheer computational power. This intervention has even more surprising secondary effects on human health and aging, suggesting that by halting the clock on certain viruses, we may be inadvertently slowing down the clock on human decay. The varicella zoster virus, which causes chickenpox and later reactivates as shingles, has been a persistent companion of our species for millennia. But new research into the latest generation of recombinant zoster vaccines has revealed an unexpected correlation. Individuals who received these vaccines showed a significantly lower risk of developing dementia in later life. One study documented an 11% reduction in dementia risk after a single dose, and a staggering 32% reduction after 2 doses. This suggests that many of the conditions we associate with natural aging are actually the long-term results of a lifetime of viral wear and tear. Viruses like zoster may act as slow-motion triggers for cognitive decline, causing chronic inflammation and cellular damage that accumulates over decades. By eliminating these infections through technology, we are not just preventing a rash, we are potentially extending the functional lifespan of the human brain. We are learning that the clock of human aging is, in many ways, a viral clock, and we are finally learning how to stop the pendulum. As we look toward the future, our strategy for pandemic preparedness is moving away from reactive treatment and toward the proactive manipulation of cellular function. We are preparing for Disease X, the unknown, highly contagious pathogen that has not yet emerged, by learning how to rewire our own internal machinery. In response, we are developing technologies like CRISPR and mRNA platforms to perform our own manual operation of the genome. We are learning to flip the immune switches that were installed in our DNA by ancient viruses millions of years ago, moving from being the products of viral architecture to being its architects. We are no longer waiting for a coincidence of exposure to dictate our fate. We are attempting to preemptively change the lock on the human cell so that no viral key can ever fit. We are entering an era of directed evolution, where the human species is beginning to curate its own genetic and immunological future. But this raises a profound question about the nature of our identity. If we are no longer being shaped by the natural world, if we are using technology to bypass the viral sieve that built our placenta and our immune systems, what will the human species look like in another 1,000 years? We have spent our entire history being moved by the invisible currents of the virusphere, like particles in a fluid. Now we are attempting to become the masters of the current itself. As we move from being the products of viral architecture to being the designers of our own biological destiny, we must consider the fragility of the systems we are building. Our survival is increasingly decoupled from our biology and tethered to our infrastructure. In a world where our immune responses are managed by AI and our infections are prevented by long-acting injectables, our technology becomes our only true skin. We have successfully halted the clock of viral evolution, but in doing so, have we also stopped the very process that made us human in the first place? And if the technology ever fails, will we still have the biological memory required to survive the next unfortunate coincidence that nature has in store? In summary, we have seen that humans are biological hybrids, sculpted by millions of years of viral interaction. From the ancient retroviruses that build the placenta to the microscopic invaders that dictated our global migrations, viruses have been the primary architects of our existence. Today, we stand at a crossroads, replacing the slow grind of natural selection with a technological skin of genomic surveillance and advanced medicine. As we transition from being the products of viral evolution to the designers of our own biological destiny, we must consider the cost of this independence. If we successfully silence the ancient dialogue between host and pathogen, do we risk losing the very mechanism that made us human? We are the legacy of a billion-year-old arms race, now holding the pen to our own story. Thank you for listening.